Hello and welcome everyone. My name is Marcia Mott and I'm the coordinator for health promotions at the University of Florida Health in Gainesville, Florida. We have a great presentation for you today on a very important topic, COVID-19. If you have any questions, please submit them using the Q&A feature at the bottom of your screen. You can type your question there and then we'll take as many questions as we can towards the end of the presentation. We have a great speaker today. Dr. Duane Mitchell is the Assistant Vice President for Research at the University of Florida and the Associate Dean for Translational Science and Clinical Research at the University College of Medicine, or the CTSI as we call it. Dr. Mitchell joined the University of Florida in 2013 and currently serves as the Director of the UF CTSI and Co-Director of the Preston Wells Center for Brain Tumor Therapy. The CTSI's mission is to accelerate the translation of scientific discoveries into practice for the diagnosis of treatment, prevention, and ultimately the cure of human disease. UF is very fortunate to have Dr. Mitchell leading the way at the UF CTSI during the COVID pandemic. Dr. Mitchell, I'm going to turn it over to you now. Dr. Mitchell, I'm going to interrupt you really quick and tell you that we don't hear you right now.
Oh, can you hear me now?
Now we do.
Thank you. All right, good morning.
Okay. Thank you.
Well, thank you for the introduction, and it is my pleasure to be able to speak this morning about the University of Florida's COVID-19 research response and some of the very innovative and exciting activities that are being pursued by our very talented faculty and staff here at UF to try to combat the COVID-19 pandemic. I will see if we can get our screen share to work here. All right. Hopefully you're able to see my screen now. And just by way of background, as mentioned, I currently in the role of the director of the CTSI and in my own primary research have focused on translational research and predominantly on taking discoveries from a laboratory setting and trying to advance them towards innovative therapeutic strategies for patients in the clinic. The last 6 months, obviously for all of us, we've been confronted by a challenge I don't think anybody really predicted we'd be faced with, but I think it's really been an example of the importance of biomedical research and the importance of translational research, where we are clearly in a time compressed scenario where we are trying to advance new knowledge into practical application for patients who are battling a very serious disease. The outline for talk this morning, I'll touch base just briefly on the background on SARS-CoV-2, the virus that causes the disease called COVID-19, and then spend the majority of our time talking about UF's COVID-19 research response. I'd like to touch on also some of the resources that have been developed here at the University of Florida to help enable COVID-19 research activities, and then we'll have time for question and answers. So for the next 30 to 40 minutes or so, I hope to give you a broad overview of some of the activities that are ongoing here at our university. Obviously in 40 minutes, I cannot possibly describe in detail the vast array of research that the University of Florida investigators are pursuing. But I think by the end of this talk, you'll get a great sense that our university is actively engaged in all aspects of translational and clinical research and basic research as it relates to SARS-CoV-2 virus. So the SARS-CoV-2 is the second novel coronavirus strain that has impacted a global pandemic. And the disease that's caused by SARS-CoV-2 is referred to as COVID-19. If we think of how quickly this virus has spread, this is a snapshot as of this morning of the Johns Hopkins dashboard, which tracks the incidence of SARS-CoV-2 infection worldwide, and we can see that all continents have been impacted by this disease pandemic, with currently approaching 26 million cases and over 850,000 deaths. This is really in striking contrast to the experience that many of you may remember with SARS-CoV-1, or what's called just SARS at that time, which struck really as a pandemic in 2003 and had a rather limited course in terms of distribution and number of people who were impacted. The coronaviruses are a family of viruses, and this is a diagram showing the structure of the SARS-CoV-2 virus, which consists of a nucleocapsid protein and RNA core surrounded by a lipid bilayer membrane, and then some key structural components, membrane protein, envelope protein, a hemagglutinin esterase, and the spike glycoprotein, which mediates viral entry into cells. It's closely related to SARS-CoV-1, which again began as a recognized pandemic in the early part of 2003, although some of the earliest documented infections were in late 2002. And one of the questions I think that has certainly struck many people is why the experience with this particular virus has been such strikingly different than the original SARS pandemic. This is an interesting article that was written in The Conversation by Dr. Marilyn Rusnick, who's a microbiologist and virologist at Penn State, describing really why the first SARS virus had such a limited lifespan in terms of spread through the human population and did not actually ultimately result in the development of the vaccine, and what's been strikingly different about this particular viral pandemic experience. Although these viruses are related and both believed to have transitioned from bats to human in terms of zoonotic jump and from animal species into humans, the original SARS virus was quite more lethal than SARS-CoV-2. In fact, estimated had about a 10% lethality within infection, but a very different course of infection, and that has played a key role in our differences in the ability to control viral spread and also in the duration of the pandemic. The first SARS-CoV-1 virus caused severe symptoms in almost all infected patients, and within a very short time span, typically within 2 to 3 days of infection, people became seriously ill. And once this was recognized as a novel coronavirus infection, it was quite easy to rapidly identify patients who were infected, quarantine them, and limit the viral spread to other individuals in the population. Also, there was a far lower incidence of asymptomatic carriers, people who were infected with the virus but did not realize it. This is in striking contrast to SARS-CoV-2, which has a much longer incubation period, thought to be anywhere from 10 to 14 days on average, and also a very significant fraction of people who are either mildly symptomatic or asymptomatic and do not recognize or are identified as carriers of the virus. This makes control measures extraordinarily difficult, and then by the time someone is identified as a SARS-CoV-2 infected individual, they have often had a prolonged contact history with others in the population that they may have had the opportunity to spread the virus to. And so we've seen, in contrast to the SARS-CoV-1 virus, which essentially had about a little longer than a 6-month course in terms of spread through the population before it eventually really died out as a pandemic, in contrast to us being well within still a rapid spread of the SARS-CoV-2 virus. This has really highlighted the importance, of course, of trying to advance research to focus on both novel strategies for detection, prevention, and treatment against the COVID-19 disease. And the University of Florida research community has responded remarkably across all spectrums related to the COVID-19 pandemic in terms of population sciences, basic science research, clinical and translational research, and clinical trials evaluating new therapeutics. I'll touch base briefly from the perspective of the UFCTSI in terms of some of the research activities that we were able to just coordinate and try to help facilitate across the vast colleges and departments that are engaged in COVID-19 research. one of the initial responses was to organize a task force to gather information about what the UF community was specifically doing in response to the COVID-19 pandemic as it relates to clinical research, translational research, education and training of the clinical workforce and translational workforce, as well as community engagement and population-based studies related to the pandemic. This task force also formed several working groups I'll touch on a little bit later that were tasked with carrying out understanding of what activities were being carried forward in various areas as it related to the pandemic, and then bringing that information back to the task force to share updates in real time across colleges and across our vast departments here at UF. This task force also included members from Florida State University, who is actually participates with us as the joint Clinical and Translational Science Institute hub, and faculty from UF Gainesville and Jacksonville. The health science leadership saw the need to also stimulate research in the area of COVID-19 research, and disseminated a rapid response pilot award program, which had a remarkable response from the university community in terms of investigators pointing their research interests and intellectual expertise towards various aspects of tackling the challenge of mitigating viral spread. There was also a recognized need for reviewing new clinical trials and studies that would require limited resources, such as COVID-19 specimens, to conduct laboratory research, and a scientific review committee was established to serve as an ancillary review in coordination with IRB review for human subjects research. The Translational Workforce Development Group within CTSI initiated a translational science of COVID-19 seminar series with featuring UF faculty who had been awarded COVID-19 pilots, and to update the research community on what their investigations were pursuing as it relates to this disease. And then there was also recognized need to develop additional resources to meet some of the specialized requirements for COVID-19 research. And then upcoming, as for those that would like to stay abreast of activities in this research space, there will be a fall symposium on October 19th, which will highlight much of the ongoing research here at the University of Florida and at Florida State in a 3-hour symposium where people will have more time to go into depth about their COVID-19 research studies. For additional updates, I just will point you to the homepage of the CTSI website, where there is a COVID-19 updates pull-down menu. And you can see that many of the topics I just mentioned have links to web pages that provide additional details that I won't have time to cover this morning. Touching base with the task force, this really was a goal to bring a wide variety of expertise from across all of the colleges here at the University of Florida to participate in discussions and create initiatives where we could amplify the efforts of UF faculty and investigators here at UF and Florida State. The task force currently consists of 43 members and was organized into 7 working groups with 11 working group co-chairs that are pictured here. This working group met weekly beginning in April through July, and now meets monthly with updates from the working groups on progress in the areas of laboratory research, clinical research, outcomes research and population-based studies, education and workforce development solutions, as well as community outreach and engagement, data science, and communications. And so this has been a way for the community to stay updated with what is going on across the spectrum of COVID-related research. And the task force also served as a gathering hub for establishing new collaborations or connecting network opportunities. The working group link is highlighted here on the website and lists all of the members there. The second need that was recognized is that there are a number of innovative therapeutic strategies being developed to try to mitigate the seriousness of COVID-19 infection. And this began a participation in the opportunity to onboard a number of clinical trials for COVID-19 infected patients. However, given that there are obviously limitations to the total number of patients that are available for clinical research studies and the acuity of care that these patients receive, particularly in intensive care units, required an organized method for onboarding clinical trials, and also making sure that the available personnel and infrastructure resources to support those studies were in place. The COVID-19 Scientific Review Committee and Peer Assistance Working Group was convened at the request of UF Health leadership, as well as the IRB, to help review protocols that come in for COVID-19 clinical trials and request specimens from these patients, and prioritize those protocols for those that could be supported here at the University of Florida. This lists the 24 members of the COVID-19 Scientific Review Committee, and again includes experts from epidemiology, infectious disease, pulmonary critical care, surgery and sepsis critical research center, environmental health and safety, and a variety of other departments and divisions within UF Health. And this committee has met twice weekly since its inception, reviewing COVID-related protocols and assessing their capabilities to be supported here at the University of Florida. Just so that people who are interested in pursuing COVID-19 clinical studies or human subject research understand that process, the committee really reviews COVID-19 research protocols and assess institutional priority, ranging from UF spearheaded research that's extramurally funded to really new or unfunded but innovative strategies addressing various questions that are important to COVID-19 research and treatment. For research that does not involve human subjects or any samples, but is interested in COVID-19 populations, such as using the integrated data repository, those are also reviewed by vice chairs of the Scientific Review Committee and do not require a full board review, but can be brought to the committee for full review if needed. And then the scientific merits and methodologies of the COVID-related research protocols are evaluated, discussed, and then ultimately scored and voted on by the committee with either approval, modification, or if it's assessed that that protocol does not have the capability to be supported, would be disapproved. This is, to date, we've reviewed over 90 protocols through the COVID-19 SRC, and is an ongoing effort to try to bring and support the best research studies that we can bring forward for this patient population that is in desperate need of innovative and new treatment strategies. A dashboard was created to try to track the different research activities that are specifically focused on COVID-19. And this is just a screenshot of that dashboard, which looks at grant proposals that have been submitted that are, from the University of Florida, focused on COVID-19, IRB protocols that have been submitted, as well as total number of patients that have been enrolled. This can be drilled down for further details as needed. I'll show some of that information just to give you an understanding of the spectrum of research activities that are currently occurring. So this is just looking at the IRB COVID-related protocols that have actually been approved to date. You can see that there's 220 total approved submissions. They're split actually evenly as the time of the snapshot between IRB 01 and 02. And then this shows those submissions over the last really 6 months. And you can see we hit a peak of protocols back in April in terms of submissions that month, but have had a steady flow of new protocol submissions each month that are reviewed and ultimately have gone on to be approved by the IRB. If you look at the spectrum of human subject research that's occurring currently, a large fraction of these studies are based on population and social sciences studies, and these include data review studies. We have a significant number of prospective and retrospective observational studies. In the interventional clinical trial space, there are currently 9 IRB approved interventional clinical trials, but this spans the full gamut of basic and bench research, data sciences, diagnostic and interventional clinical trials, as well as observational studies and quality improvement work. And so you can see the UF community is really engaged and focused on COVID-19 in a variety of dimensions, both within the laboratory setting as well as out in the community. This is a listing of the current interventional clinical trials that have been onboarded for patients with COVID-19 and their respective PIs. And these include multi-center randomized controlled trials that we're participating in as national participants being supported by various institutes and the Health and Human Services, and also innovative investigator-initiated trials that were developed here by UF investigators, example by Dr. Freddie Kay, who was studying the potential to interface with the androgen receptor pathway in males as perhaps a way of disrupting COVID-19 infection in male infected patients at risk. And so there's a interesting gamut of different treatment strategies that have been onboarded. We're currently looking at small molecule inhibitors, anti-inflammatory agents, as well as monoclonal antibodies. And I'll touch base a little bit on some of the new treatment strategies that are even under development by research labs here at the University of Florida. One of the goals of the Scientific Review Committee is each week we get updates on the enrollment across these trials, the spectrum of clinical trials and the patient populations that are eligible for these trials. And as they're examining new studies, looking for gaps in coverage for clinical trial options for patients, and also consideration of inpatient as well as outpatient studies. The UF Health leadership also recognized that since this was a new threat that rapidly emerged, that many people obviously did not have funding focused on SARS-CoV-2 or COVID-19, and to catalyze the university's research efforts, issued a pilot awards, RFA, in the month of April. This was open for 30 days, in which we received 116 applications from the UF community for new projects focusing on COVID-19 research. This was reviewed, went through a scientific review process, including topic expert reviews and statistical reviewers, and ultimately resulted in 19 awards and $1.38 million of awarded funds. And this has supported a diversity, again, of COVID-related research. The recipients span 8 different colleges and 18 different departments, and are supporting research, as you'll see on this list, that ranges from the immunologic phenotyping of patients with COVID-19 infection in a hospitalized setting, the development of new or screening of effective antiviral strategies, telehealth and doctor-patient communications in cancer patients was co-supported by the UF Health Cancer Center, a data informatics study to look at developing models for actionable clinical and administrative decision-making. This was supported in collaboration with the UF Informatics Institute. And then I won't read through the whole list, but even development of novel vaccine strategies within UF laboratories using RNA-based nanoparticles. And so there's a huge breadth of research that's being undertaken by the UF community. And obviously, out of 116 proposals, there are clearly more meritorious efforts moving forward than we have the opportunity to support. But it's one of the nice things to note is that many of those proposals, including some funded through this pilot mechanism and others that were not, have gone on to receive extramural funding to continue those research efforts here at UF. For people who are interested in getting details about these pilots, I'll point you to the web link that summarizes where you can get details about each of the awards. This is just a snapshot of some of the specialized facilities. This is Dr. Michael Norris in the BSL-3 facility here at the Emergent Pathogens Institute. He and his colleagues, John Lednicki and Christopher Volpe, are supported with a CTSI pilot award that's looking for novel targets for the SARS-CoV-2 infection using a gene disruptive technology, and working actually with clinical isolates of SARS-CoV-2, which does require BSL level 3 facilities that UF maintains both here as well as on other campuses in Orlando as well. And so there's a huge amount of expertise, obviously, in the Emerging Pathogens Institute, focused on not just the biology of this infection, but the scalability of rapid detection technologies. And Glenn Morris has been instrumental, actually, in helping establish some of the rapid screening approaches to testing for SARS-CoV-2 infection in the general population. And so a lot of really innovative and exciting work that's occurring from basic virology as well as screening and detection. To really make the community aware of these efforts, Tom Pearson and Wayne McCormick and colleagues in the Translational Workforce Development Group and education group, started this Translational Science of COVID-19 seminar series. We've had 6 of the recipients that have presented their work thus far, and those recordings also can be found with links on the CTSI website. And we'll continue with that series this fall semester so that people can stay updated on the work that's being supported through those pilot awards. There's also been really outstanding opportunities for UF's leading expertise in population-based research and wide established integrated clinical data networks that really have been built over the last decade by most notably Dr. Betsy Shankman and David Nelson in developing the One Florida Clinical Research Consortium and Data Trust. Many people are familiar with One Florida. If those who have not been introduced to this is a network of clinical research, clinical treatment and research sites throughout the state of Florida that have been organized into a research consortium with existing data sharing agreements, consolidated in a single IRB activation mechanisms for research studies that's housed administratively here at the University of Florida, and really allows for community-based pragmatic clinical trials and large data studies on a vast population of patients to be pursued in an efficient fashion. Currently, One Florida network covers about 40% of the state population, greater than 15 million patients, and has data registries and data collection, active data collection on patients from all 67 counties within the state of Florida. With the need to rapidly stand up both interventional clinical trials as well as acquire data on patients who are impacted by COVID-19, the One Florida Research Network has served as a key resource for clinical trials as well as data and informatic research. One of the early clinical trials looking at hydroxychloroquine as a preventative or prophylactic strategy in healthcare workers called HERO HCQ was deployed through the One Florida network in record time, in about 2 to 3 weeks from protocol receiving the protocol to actually activating and going live with enrollments. And this was really a testament to a decade of efforts that have been put in place building this network and resource, as well as the really rapid response of the IRB and the Office of Clinical Research, understanding the urgency of COVID-19 research. Additionally, there's a national effort being spearheaded by the National Institutes of Allergy and Infectious Disease, focusing on trying to understand the immunobiology of SARS-CoV-2 infection, with the hypothesis that patients who progress and have more severe disease may likely have a differentiating immunologic response compared to those who successfully resolve infection. And this national effort to try to comprehensively immunophenotype 2,000 patients with COVID-19 infection has been initiated throughout the United States, but was approached to the University of Florida to see if the One Florida network could be utilized to give coverage for patients throughout the state. I'll talk a little bit more about Dr. Atkinson's work in that effort. And then as high priority national clinical trials have been erected, looking to enroll large numbers of patients in a short period of time. Again, the One Florida resource and network has been tapped into to really provide a nimble mechanism for deploying clinical trials throughout the state, and has become the vanguard site for many prominent national clinical trials. This is a heat map of the impact heat map of COVID-19 infection, and a highlight of the sites throughout the United States that are engaged in this immunophenotyping research. And as the pandemic, as many remember, really had high concentrations on the West Coast and Northeast at the early stages of the pandemic when this effort was initiated. But then we've seen the hotspots for infection transition over time more into the South and Southeast and Southwest. And so as the NIAID was looking to try to enroll patients, they approached Mark Atkinson, who's had long-term funding from the NIAID through a P grant, about whether they could participate in this immunophenotyping effort here in the state of Florida. And the One Florida resource was able to respond with demonstrating not only did we have ongoing research looking at immunophenotyping that was being supported by our pilot awards here at UF, but also had pre-existing relationships with sites throughout the state of Florida that would allow for the demographically and geographically diverse enrollment of patients for this study. And so again, in relatively short period of time, they were activated to participate as the site for the state of Florida in this comprehensive immunophenotyping research. If you look at the efforts as it's related to COVID-19 therapies, we are also engaged in advancing research across the spectrum. There have been, three main therapeutic targets, as well as a number of other emerging strategies. A lot of attention has been given to trying to suppress the significant and severe inflammatory response, particularly pulmonary inflammation that occurs with COVID-19 infection, and approaches to try to mitigate this inflammation have really had modest effect in severely ill patients, with the notable exception of a few medications that do shorten disease course. There's strategies to try to directly inhibit viral replication, and those are being currently evaluated in ongoing clinical trials. And then there's some attention, although not as much, and I'll touch base on this, to also dealing with another spectrum of the COVID-19 infection, and that is the development of significant immune suppression in patients who go on to develop advanced disease. There are also strategies focusing on some of the significant complications that this infection ensues, such as thrombotic events, and looking at anti-thrombotic thrombolytic therapies. And so there are really a wide variety of approaches being pursued, and researchers are engaged in looking at the mechanisms in all of these fronts. I'm going to talk about some of the novel work just because as an immunologist, I happen to have a little bit of bias and understanding of some immune mechanisms, and some work that is ongoing with investigators here at UF, really focusing on novel aspects of the immunologic response in these patients. So this is a recent publication with colleagues from Washington University in St. Louis, as well as here at UF, most notably Scott Breckenridge and Link Moldauer in the Sepsis and Critical Illness Research Center here in the Department of Surgery. Link and Scott have studied for some time the spectrum of immune response that we see in patients with severe sepsis, where initially significant inflammation and perhaps over or hyperactive immunologic response is certainly a component of the disease spectrum. But what you also see is this progression towards an immunosuppressed state in patients with severe illness. Taken from this publication is showing a subset of patients with COVID-19 infection who either survived or did not survive after admitted to the intensive care units. And this is looking at their absolute lymphocyte counts, or the number of T cells and adaptive immune cells that they have in circulation that are potentially the effectors that can actually mediate the elimination of virus from the body. And you can see that having a low lymphocyte count, or lymphopenia, upon admission into the intensive care unit was really a poor associated with mortality with COVID-19 infection. And this has been seen in other studies and published by other groups, that this depressed T cell response, or absence of T cells, is associated with inability to resolve the infection successfully. They went further to show that if you look at the function of these T cells, so what you're looking at in these circles, each one of these dots represents an immune cell that is secreting interferon gamma, or is active when it's challenged with an antigen. And so the more spots, the more active, larger number of T cells these patients have that are capable of mounting a response. You can see that in healthy controls and in non-critically ill, non-septic patients, you can see a robust T cell response. But when you get to patients that either have critical illness with sepsis or with COVID-19, we see this marked diminution of T cells that are capable of mounting an immunologic response, underlying immune suppression as being a significant component of this disease. Link and his colleagues also published a nice article in the Lancet Respiratory Medicine, really highlighting some of the differences that we see with COVID-19 infection and polymicrobial sepsis. And one of the notable features is this early severe lymphopenia that results in prolonged immune suppression and uncontrolled viremia in these patients. And so there's been an emerging hypothesis that perhaps one of the real challenges with resolving these significant infections is actually the inability to mount an effective adaptive T cell response. This is, or the loss of the ability to maintain an effective T cell response. This is an article looking at patients from Wuhan, China, a large number of patients, again, underscoring similar findings of this loss of T cells in significantly ill patients. And what you're looking at in these graphs is actually the expression of markers that are associated with T cell exhaustion, or being the T cells that are there being incapable of responding. And you see an increased expression of these exhaustion markers, PD-1 most notably, on severely ill patients. And so this hypothesis that in addition to perhaps what we call this innate inflammatory response in the lungs, perhaps the failure to clear virus or resolve infection may be due to this T cell response that is either not maintained or lost, either unable to be mounted or lost during the course of COVID-19 infection in some patients. And so an alternative strategy in thinking about combating this virus is trying to augment the adaptive T cell response to SARS-CoV-2 virus. And this is some work that our researchers within the Brain Tumor Center that have been focusing on immunotherapy in cancer patients for many years have begun to collaborate with investigators here within the Sepsis and Critical Illness Center to explore the hypothesis that strategies that perhaps could reactivate an effective T cell response, particularly, could be effective in critically ill patients with SARS-CoV-2 infection. This particular research has been led by Dr. Flores, who's an assistant professor in the Department of Neurosurgery, and she has made some novel discoveries that stem cells from the bone marrow combined with PD-1 blockade, blocking one of these immune exhaustion pathways, could lead to a unique ability to synergistically reactivate or reawaken T cell responses in preclinical models of advanced cancer. I'm just going to show some data from their published work in Nature Communications, as well as a series of other papers showing in lymphopenic, these are now preclinical studies, but under conditions of lymphopenia, the transfer of hematopoietic stem cells actually causes the production of cytokines that give rise to T cell growth. And if you now treat with a PD-1 inhibitor combined with stem cell transfer, you actually get a rigorous repopulation of T cells in the periphery in preclinical models. This suggests that there's a lot of mechanistic data that I won't have time to show, but essentially there's a synergistic interaction between blocking this exhaustion pathway and providing stem cells to regenerate very effective T cells in the periphery. Last data slide along these lines. This is gene expression, looking at the top at markers of T cell exhaustion and looking at the bottom of markers of T cell activation. And what you can see is that, again, with this combination of stem cells and checkpoint blockade, we get this unique activation now of T cell responses and a loss of this immunosuppressive markers that are expressed in the untreated or single treated hosts. And so we had been pursuing for some time this novel combinatorial strategy to potentially deal with T cell exhaustion and immune suppression in patients with cancer. And in fact, had just recently submitted to the FDA requests to start clinical trials using this approach in immunosuppressed patients with brain cancer. And in discussion with colleagues, most notably Link Moldauer and Scott Breckenridge, as well as with John Wingard in our stem cell transplant group that he leads, began to realize that there was an opportunity to potentially address immune suppression and exhaustion in patients with lymphopenia using this novel strategy. And so that has resulted in a collaborative effort over the last several months to adapt our FDA investigational new drug approach to dealing with this patient population, and a clinical protocol concept that has been developed with these colleagues. And we're still gathering additional information to look at advancing the potential of advancing this strategy into a novel first-in-human clinical trial here at the University of Florida. Underlying this has been some studies that, again, colleagues here at UF already participated in, looking at checkpoint blockade, PD-1 blockade in patients with sepsis. These were not studies in COVID-19 patients, but prior studies looking at this strategy of trying to deal with immune exhaustion in patients with sepsis. And so there's already an existing human clinical experience, both with doses of PD-1 blocking drugs, as well as the spectrum of safety profile within these patient populations. And so we're encouraged again by the opportunity to potentially collaborate with many of the innovative investigators here at UF to develop new strategies in trying to combat this very challenging disease. The last couple of minutes, I will touch on resources that have been developed to help support COVID-19 research here at the University of Florida. I mentioned to you that there's we have an integrated data repository that has a COVID-19 cohort where people can extract clinical data on COVID-19 tested subjects for discovering outcomes related studies or biomarkers that are clinical markers that are associated with differential response to treatment. The One Florida data network I mentioned to you. The other two that I'll touch base on is there was a recognized need for people to be able to access specimens from COVID-19 tested subjects. And yet these patients obviously are in a clinical scenario where gaining access to them by researchers, particularly in the laboratory setting, is very challenging or limited. And so the CTSI Biorepository, with the help of Dr. Mike Claire Salzer, Amir Abu Hamza, who directs the CTSI Biorepository, and Mark Brantley, worked with the IRB to erect a COVID-19 biorepository. This biorepository has to date collected samples from over 1,100 subjects and 34,000 samples, with a range of different samples that can be utilized to support COVID-19 research listed here. There is also a web link for further details for researchers who are interested in accessing these specimens in terms of the process to be able to retrieve them from the biorepository. This is an ongoing prospective effort in order to provide the requisite resources to conduct investigations using these limited biospecimens. There's also been a shift in the focus of clinical treatment to not just dealing with patients who are hospitalized and have severe illness, but as we've gained a better understanding of who are those patients who are most at risk for developing severe infection, to try to intervene earlier in the course of COVID-19 in the outpatient setting in order to prevent progression to hospitalization and to limit the demand on hospital resources. This interest in conducting clinical trials in the outpatient setting presented several challenges, one most notably being that patients who are well enough to recover at home and don't need hospitalization are oftentimes not brought back to clinic for follow-up. As you can imagine, during the acute course of an infection, we're trying to quarantine or limit exposures. And so there was really not a good opportunity to bring research subjects back for longitudinal follow-up. And so this prompted the need to examine various options for a safe environment for longitudinal studies in mild to moderately ill patients who do not need hospitalization. And that has resulted in investing in a resource to provide a mobile clinic. And so this is just an artist rendition, and don't hold me accountable if it doesn't look exactly like this, of a clinical research vehicle that would be used. This is a mobile clinic that has been acquired by University of Florida. This is how it actually looks in its current state, and has an interior to this mobile clinic, laboratory facilities, as well as two clinical rooms that can be used for patient examinations, adequate and appropriate ventilation, and obviously the ability to visit subjects away from other patient populations. And so we're excited about being able to bring this resource on to make outpatient COVID-19 studies and longitudinal follow-up something that can be done in a controlled and safe environment. And this is just an example of the types of research that are being developed and being onboarded as we gain better understanding of the pandemic. So in summary, I hope I've convinced you in obviously very broad strokes that UF researchers are engaged in pioneering efforts in all aspects of the COVID-19 research. That includes prevention strategies with novel vaccines, therapeutics with novel treatment approaches, basic virology, as well as uncovering new targets through mechanistic understanding of viral infection, as well as population and data science-driven analyses that are coming up with better strategies to both diagnose, treat, and prevent the COVID-19 pandemic. These efforts will have far-reaching impact. They already have had and been recognized for their national prominence and capacity to contribute significantly to our national research efforts, but will continue to yield dividends in providing important data that should lead to the resolution and recovery from the global pandemic. So I appreciate your attention, and I think the remainder of the time, I'll stop the screen share, is to be available for questions.
Okay, that was great. It's really interesting about the mobile research vehicles. So we have several questions that have been coming in, so I'm going to go ahead and start going through those. One of the first ones is, do you think that we can push and perfect a personal at-home test to help empower people to know where they stand and figure out how to move forward?
It's a great question, and there are, so the short answer, and I'll by disclaimer give my background. I'm not a virologist or microbiologist or infectious disease specialist by background, but I can certainly have seen the wide spectrum of activities that are already occurring in the diagnostic or the testing space, including, as you described, rapid and in-home testing kits that are actually available, and some have been made. Some have even gotten what's called an emergency use authorization from the FDA. I think and so those strategies are being developed and continually refined. To my knowledge right now, the tests that are available, I believe, allow people to send a specimen in that still is tested in a laboratory setting. There are also tests that are based on trying to have sort of basically a colorimetric change, where you would provide a specimen, whether it be saliva as an example, and literally just look for a particular color change on a strip to indicate whether it was positive or negative for viral infection. So these are in various stages of development already and being tested and evaluated for their sensitivity and specificity. And so yes, I think that you'll see in the coming weeks and months, a wide variety of new testing strategies coming forward. Okay,
thank you. Has UF conducted research on treatment protocols for home patients or in-home hospital patients?
In-home, so short answer is yes. We have on board, we have both inpatient and outpatient clinical trials that have been onboarded and approved here at UF. The majority of those in the outpatient setting currently still are on patients where they've been enrolled here, seen at a point of care, and then diagnosed with being recently infected with SARS-CoV-2, and then are being given treatment, follow-on treatment in the outpatient setting, so they're not sick enough to need hospitalization. I'm not aware of a clinical trial yet that is specifically focused on people that are already, for instance, on in-home hospital care. But what is being evaluated, and in fact, we're in ongoing discussions right now, is onboarding studies that continue to allow patients to be monitored more aggressively at home. So using in-home monitoring, continuous monitoring, things like being able to capture oxygen levels and a lot of information about how patients are doing using remote sensors. And so there's a great interest in being able to bring treatments and provide care in the home setting. One, it's better for, obviously for patients to have limited access to a major treatment center, but two, it also, to the degree that we don't have to bring people into a hospital and can still deliver adequate and appropriate care at their home and monitor them safely, that would be a huge advance, not just for clinical trials, but for clinical management. And so these are efforts that are actually currently underway, and in fact, evaluating studies that are using those approaches. They just haven't been opened yet for enrollment.
Okay, that's going to be important in the future, I think. So, let's see. Will there be any COVID-19 vaccine trials at UF? If so, if people signed up online to be a part of such trial, would they be contacted? And how soon would this happen, possibly?
Yeah, it's a great question. There's obviously a lot of interest in participation in vaccine trials. The short answer is yes, we have. We're very interested in looking at vaccine trials. I believe the UF Health Jacksonville site has actually been identified by one of the vaccine studies as a potential. They've had outreach to be activated as a site for vaccine participation, and we're continuing to look at other vaccine studies as they come on board to make them available. As you're probably aware, and I think alluded to in the question, there's a number of different ways that people are able to get information about vaccine studies, and some of them have made direct outreach where you can, register online at the individual level of expressing interest. Those are not, to my knowledge, coordinated centrally through UF or certainly up through CTSI. So exactly how the follow-up mechanisms from those studies would work, it's probably due to whoever the sponsor is that's actually supporting those registration sites. How we would onboard a clinical trial, though, that was a vaccine study here is typically there's an investigator that's leading that study, but then when it is opened for enrollment, the participation in that study would be for patients who are eligible, subjects who are eligible, healthy volunteers who are eligible. As an example, the HERO registry for healthcare workers is a registry that anybody who's involved in basically clinical care of COVID-19 patients can register, or is interested in potentially being contacted for future vaccine studies can also register. So there's a number of mechanisms where people can express their interest, but as trials are open formally within the center, we would push that out in terms of advertising the enrollment for those studies.
Okay, thank you. Do we know how or where the first SARS virus originated?
So great question. It is believed that both SARS-CoV-1 and SARS-CoV-2 originated in bats. That is the current understanding. And both of these in pandemics, if you will, or took hold when that virus spread from the bat species to humans, the first human contact, both believed to have occurred in China. The exact mechanism of how that initial infection may have occurred, I think is still being evaluated, but essentially, as people come into close contact with more species, whether it be through incidental contact, through studying animal species, and just expansion of populations into areas where these animals live, then there are a variety of means by which the virus can be transmitted from the animal species to humans. And the virus obviously has to be capable of growing both in its original species, in this case the bat, as well as in humans. And so the same species is believed to be the origin, but exact mode of transmission I don't believe is well delineated.
Okay, thank you. What is your recommendation about the safety of immunocompromised at-risk seniors taking part in any approved vaccine before it completes a phase 3 review?
It's a really great question. And so, for anybody that's participating in, and I'll speak to it in clinical research studies in general, and then touch it. There are whenever we're conducting clinical research, including if it's a vaccine study or otherwise, there's a process obviously of explaining through informed consent that the complete understanding of the safety and the effectiveness of the treatment is not at hand when the clinical trial is being conducted. And so the risks and benefit to a person participating in a study really is something that has to be a thoughtful consideration and discussion, ideally with their both physician, whoever's conducting the study, the principal investigator and the research team, as well as their own physician and family and others that may have input, to try to reach a decision of what they believe is their individualized risk-benefit ratio. Vaccines, generally speaking, are one of our most widely distributed forms of treatment, one of our most widely effective forms of treatment in terms of medical advances. I'm an immunologist, but if we just look at treatments that have been effective in eradicating diseases, I would have to say prophylactic vaccines stands out to me as probably the modality that's had the largest impact on eradicating disease in the human population, and relatively speaking has a good safety profile. That said, that's generally speaking. There's always risks that can be associated with any treatments, and that would have to be something that a person would have to have a thoughtful discussion as to whether they believe that participating in a vaccine study offered more potential benefit versus potentially self-isolating and trying to remain limiting contact. And I think that's not something I would take lightly, and I would just have that discussion with both the care provider as well as the study team to make that decision. I'll add one more note to that, though, with respect to the immunocompromised state. Many of the vaccines that are being developed currently are not using the virus itself. Some vaccines and some vaccines that we still use today are what are called attenuated or inactivated viruses. They are the original virus kind of modified in some way or inactivated in some way to be less aggressive and form a mild infection, which can induce immunity. Many of the vaccines that are being developed right now for SARS-CoV-2 are not modified from the forms of the virus, but are actually subunits or parts of the virus. So in terms of the risks of getting a significant infection, as an example, from that vaccines, those are certainly largely eliminated when you're not using a inactivated or attenuated, what they call, form of the virus. And that's one big differentiator, but it's still an important discussion in terms of the overall risk benefit.
Okay. Can you tell me how many strains of SARS-CoV-2 there are and how fast they're mutating?
I don't know that information firsthand. That's a great question. There have certainly been described different viral isolates, predominant viral isolates in regions. For instance, if you look at what originated early detection in Wuhan, China, compared to Europe and the United States, there have been some divergence, if you will, in terms of the viral sequences that are most prevalent and detected in the population. And viruses in general, because of their rapid replication, can mutate and frequently do mutate. But we have fantastic virologists here at the EPI Institute that keep up on top of exactly the number of strains or the number of, I would say, variants that are seen. Because one of the things that has to be determined is when you see differences in the viral sequences, have you detected a distinct strain, or is it a variant that has, for one reason or another, become more prevalent in a population? So as you can imagine, there may be several viruses in one population. If somebody travels and they happen to be harboring one, they may travel to another region and become the founder, if you will, of the infectious spread in that region. So if you were to look at the two viruses or the predominant virus in one area versus another, you would see differences, but that wasn't necessarily because that virus mutated and changed to become this new strain. It may have been what's called a founder effect. Somebody migrated that happened to be carrying a particular type of the virus. So you can see different variants in different regions, and that can come about both from true mutations as well as through variants that have differential a way that they spread through a population in different areas. And there's a lot of information being gathered on that, but that's not an area I can give you the specifics on.
Okay, gosh, there's so many variables and it's amazing. It's kind of boggling to keep up with. So are there any studies of specifically of the transmission of the disease that you could talk about? Any novel results so far?
Studies on the transmission of disease. So yes, and I'm not. I'll try to answer it and hope that I'm hitting on what the person asked the question wanted to get at.
Yeah.
So there's interest in understanding how, the molecular mechanisms by which the viral spike protein, which is believed to be one of the major protein for allowing viral entry through the ACE2 receptor. This is a receptor that's widely expressed on a lot of different cells in the body. And so we know that the virus is capable of viral entry into a variety of tissue cells and organs. And what there's a lot of study on is both, can you understand specifically what parts of the protein are important for viral entry into cells? That can give strategies for trying to block those interactions, whether they be with specific antibodies or specific drugs. We have researchers who are looking for molecules that will disrupt that interaction between the major protein of the virus that's involved with entering into the cells in terms of transmission. There's also a lot of study being done on what happens after the virus gains entry into the cell. So just getting into the cell is obviously important, but there are many subsequent steps that are necessary for the virus to take hold, replicate, and spread. And so at the molecular level, understanding those steps can lead to new ways of disrupting some of the downstream requirements for the virus to continue to productively spread. You said transmission, and so that can occur, of course, then you're talking between people. There's a lot of interest in trying to understand what are the major modes of transmission. If you've kept up with any of the headlines, you've seen, droplets, respiratory droplets, you've seen, airborne transmission, you've seen a lot of different terms. And what's really people are focused on is we know that the virus can be detected from someone who's shedding virus at certain distances, for instance, and for certain periods of time. But what is the primary mode by which others actually get infected is really important when you're trying to understand how do you control viral spread in a population. So is it simply just having any virus present in the environment, or is most of the transmission really occurring between people who have close contact for certain periods of time? Studying that and continuing to study that is really important because it influences what policies or what practices that we can implement to best mitigate the spread. And so that's a really an ongoing area of research, and a lot has been learned, but it's still an area of intense study. And then you've seen studies that are actually looking at, and we've had, again, investigators that are participating here at UF in understanding how effective are different types of barriers at preventing the virus from being detectable. So if you look at different types of masks compared to not wearing masking, or whether you're coughing or sneezing or just talking, are there different most effective methods for reducing how much virus can be released into the environment? So from molecular to person-to-person transmission are all active areas of research, and we have investigators here at UF that are actually engaged in every one of those levels of investigation.
Wow. So many things happening. Wow. So we have a question. We have someone watching from another country, Bangladesh, and they want to know, what are the alternative options for COVID-19 treatments in a developing country?
That's a great question. So, as you're probably aware, much of the care that we are giving to patients routinely right now with, say, significant COVID infection is what we would call supportive care, trying to essentially support their vital organ systems to not succumb to the complications of the virus. We're still actively looking for really effective treatment strategies to help resolve the viral infection or clear virus from people who are infected. Some of the investigational agents that are being looked at, such as monoclonal antibodies or others, are resources that are treatments that can be resource intense. I do think that there's a few things that are worth looking into for sure. One is that many companies, as well as philanthropic organizations like Gates Foundations and others, are well aware of the need for vaccines, therapeutics, and detection technologies to be made available globally. It's obviously been a challenge scaling up to meet this need for developed countries, developing countries as well. But there's a recognition of the importance of doing that. So some of the treatments, they have pathways to make some of these drugs available globally to other countries. And then the treatment strategies, I think certainly prophylactic vaccines are something that there's a huge focus on being able to make both the trials and manufactured vaccines globally available. When you think about cost-effective methods and ways to focus, if we really look historically, the treatments that have been the fastest from sort of testing to deployment are really repurposing existing drugs. If we can find existing drugs that we already know a lot about their safety profile and we already know a lot about their pharmacology, if we can find effective drugs that are already sort of out there in the marketplace, if you will, that work, those can be more quickly and cheaply deployed because much of the cost of the development of the drug has already occurred. So I think screening, and this is still an active area, screening rationally for drugs that might make sense for possibly being able to disrupt the viral life cycle and or the inflammatory cascade. We also think that the immune suppression is worth looking at, but looking at existing drugs that maybe have some efficacy and testing those in trials is also, I think, a rational strategy based on resource consumption.
Okay, kind of along the lines of that, how do you feel about the trajectory of this virus and a time when we can go on more normally with everyday activities?
Yeah, I think it's the question we all ask ourselves every day. When are we getting back to normal, and what is that normal going to look like? As a scientist, I'm very data driven. I run a research laboratory. I think I like to let the data guide my sort of rational thinking, but I'm also an optimist in that I believe that solutions through the concerted effort that we have will be found. We've never had as many people, I think, intellectually engaged on solving a single problem in science that I can think of, at least in recent times, as we do right now. So the not just being an optimist, but I say that, we don't have any benchmark to compare how quickly can we achieve something when you have the entire global medical and scientific community and population all agreeing that this is a single and very important goal. So I am hopeful that means we will see advances and breakthroughs much faster than we would have otherwise, because there's such a concentration of effort going into battling this pandemic. That said, I'm encouraged by the same things that others are seeing. I think we hope we'll have vaccines and late stage testing going into the end of this year. I am as hopeful as anybody else that we'll have effective, therefore, vaccines widely available through next year. I also think treatments that mitigate, we're seeing that our physicians and nurses and healthcare workers have been remarkable at just taking better care of patients as we begin, being able to get people through this difficult disease as we've learned more and gained more experience. So patients, it's still a very significant and severe infection, but we are seeing that, the ability to get people through this is getting better and better and better. And so I think effective treatments that lower the severity of the disease such that fewer people have to end up in the hospital, fewer people in the hospital end up on ventilators, fewer people that, do need that level of support, don't recover, but actually do recover. I think that there's great hope that we'll have some treatments that will reduce the severity so that while we're waiting for something that is very effective at protecting the whole population, we'll be gaining confidence that we can ideally identify those patients most at risk and get them through this disease with far less significant morbidity and mortality. So I'm optimistic that next year we will see those changes coming to fruition. Don't have a date on when I believe we'll be back to normal, but I do think the medical community and scientific community will respond to meet this challenge.
Yeah. Okay, another question that we have, are there any plasma treatments or trials going on now with plasma in the Gainesville area?
Yes. So I think what you're referring to is convalescent plasma, and that is where a person who has recovered from COVID-19 infection and has developed antibodies naturally against the infection. It's one of our natural ways the immune system fights viral infection is producing specific antibodies. That we collect the plasma from those patients and then administer enrich the plasma for those antibodies and administer that enriched plasma to patients who have been diagnosed with new infection. And the hope there is that by giving a large dose of already preformed antibodies against the virus, you now slow or prevent the spread of that new infection in the host that's received the plasma. So we do have an ongoing clinical trial here at the University of Florida using convalescent plasma. Dr. Merck is the principal investigator of that study. And it's a treatment that we're wanting to gain information and comparing how people who receive that treatment do compared to patients who do not. And the hope would be that we would gain enough information to know the effectiveness of the convalescent plasma and which patients really just seem to derive the most benefit from that. This is a national study that we are participating in here at the University of Florida.
Yeah, I hope that goes well. I know someone who just donated theirs yesterday, so exciting time. So here's a pretty timely question. Can the pneumonia vaccine, or at this time of the year, a flu vaccine, can it help prevent or lessen the severe effects of COVID?
Great question. And I can answer that by saying we don't know, but there is actually a lot of interest in this potential of some kind of cross protection. And when I say interest, what I mean is that people have looked through one of the advantages of having clinical data. If you collect clinical data from people in a way that it can be what we call retrospectively analyzed, or you can look through that data, there have been some suggestions in looking through the data that certain types of prior vaccination, patients that have received certain types of prior vaccination may, and the operative word there is may, because when you're looking retrospective, you're really looking for associations. They're not proving any kind of cause and effect, but they're saying that in this data, there's an association between maybe people who had less severe disease and perhaps received a particular type of vaccination in the past. So there is data that is suggestive that there can be some potential cross-protective, if you will, mechanisms from vaccines. Not well understood in terms of whether that is really an association that's not related or whether there's something there, but a lot of interest in studying this going forward. So I don't know that, I think we would recommend, people recommend getting the vaccines that are recommended for protection against the infections that those vaccines have been proven to be effective against. So what I would say is that there is some science and some scientific interest, though, based on some observations of studying whether there are any cross-protective benefits of other vaccines.
Okay, yeah, it's getting to be about that time where we all have to start thinking about that again, flu shots and all those things that happen with fall and the cooler weather. So, well, that's about all the time that we have questions for today. I want to thank everyone for joining us, and I appreciate you tuning in. We have recorded this webinar, and we will send a recording link out with to each one of you after this event, and then it'll be posted on ufhealth.org/wellness. And I'd like to thank you. Thank you so much. And Dr. Mitchell, say?
Oh, no, thank you. I appreciate everybody's attention and great questions. And remember, wash your hands, wear your mask, get your vaccines. And,
also, when you leave today, a survey will pop up to let you know, to let us know what you thought about things today, or provide any other feedback if you forgot to tell us something, or give us a suggestion. I appreciate it. And thank you again so much. Bye-bye.
All right, thank you.